Hello and welcome dear viewers to the another informative topic on scale up and technology transfer for pharmaceuticals. So in this video we will discuss the third stage of the research and development or product development in the pharmaceuticals, and scale up and technology transfer is the very important stage in formulation development and technology transfer department, and it is a very important step for the product development as it involves the increase in batch size, that is scale up, and the technology transfer from R&D to the manufacturing site. So the phases of drug development are or drug product development are mainly this is regarding the formulation and not regarding the API. So we will discuss only about the formulation development. So the phases of drug product development involves research and development, then scale up and technology transfer, then validation and commercial production of the developed formulation, and scale up and post approval changes. These are also known as SUPAC, and there are SUPAC guidelines. So in US it is known as SUPAC or scale up and post approval changes, and in Europe it is known as variation. Then comes to the life cycle approach. So these are the four important stages. Out of that, the research and development involves the lab scale design and development of the prototype formula and the prototype process. Then this prototype formula and process moves to the scale up and technology transfer stage, in which the batch size is increased as per the regulatory requirement for product filing. So that is around 1 lakhs, and always the batch size is taken above 1 lakh, like 1.5 lakhs at least batch size is taken for scale up and technology transfer. This involves product development lab for the scale up and technology transfer, that is also called as TT in the pharmaceutical industries, that is technology transfer, exhibit batches and process qualification batches. So the entire step involves the scale up and technology transfer. Then validation and commercial production. Once the product get approved after completion of the exhibit batches, then this validation is done and commercial production is started. Then if there is any change to the composition or manufacturing process or manufacturing site or equipments or anything regarding the finalized commercial product, then scale up and post approval changes are done, that is involved into the stage of life cycle approach. In the scale up and post approval changes, the equipment changes may be there, minor changes or major changes will be there. So as per that requirements, the SUPAC guidelines are applicable, and those SUPAC guidelines are used. Then research and development, lab scale design and development of the prototype formulation. So the research and development involves selection of APIs, excipients, machine change parts like tooling for the tablet and dosing disc for the capsule formulation mainly. Also the change part according to the product specific are selected and developed, and those are shared with the manufacturing plant for successful technology transfer. Then selection of the manufacturing process. So whatever the manufacturing process is selected, that is based on the prototype formulation, and the manufacturing process is selected based on the equipment availability on at the manufacturing site. Also this manufacturing process is a prototype process at the R&D, and it involves the selection of manufacturing process based on the composition and the reference product. So as per the reference product design, these APIs, excipient, machine change parts and manufacturing process is selected. Then pre-formulation and compatibility studies are done to check any incompatibility of drug substance with the excipient or packaging materials. Then lab scale stability is performed. The lab scale stability is performed at 4075, 3065, 3075 and long term. Then, as per the product requirement, pilot bioequivalence studies is done with the lab scale batches so that there will be additional confidence of passing the pivotal bioequivalence. Then final prototype with scalable manufacturing process is made ready, which passes the pilot bioequivalence and have a satisfactory lab stability data and a prototype manufacturing process. So now this phase gives the final prototype formula for scale up and technology transfer. Then comes the stage of product development lab TT, product development lab, that is PDL or TT, and exhibit batches. It is also known as process qualification batches, where the robustness of the product is tested on 3 exhibit batches. So product transfer from R&D to production, sometimes separate PDL lab is there, or sometimes separate TT lab is there, or TT area is there, which is approved by the regulatory authorities. Then transfer of the knowledge and technology. See, only the product transfer is not sufficient in the pharmaceutical field. The requirement is that the knowledge gained in the research and development regarding the product and process, that should be transferred and shared with the TT team, that is the people who are involved in the tech transfer team or PDL lab, so that there will be a successful transfer of the technology. Then scale up batches are taken. Based on the scale up batch results, exhibit batches are taken. Exhibit batches are also called as pivotal stability batches. Then out of these batches, the batch is selected for the pivotal bioequivalence study. And after having a satisfactory stability data and pivotal bioequivalence data and the robust manufacturing process, the regulatory filing is done. And the regulatory filing is proceed with approval. Approval from the regulatory authorities like the USFDA or EMA or any other as per the filing of the product. Then we should understand what are the criticalities into the tech transfer and what are the opportunities. And opportunities are there to make the tech transfer successful, so that the product development stage will not be lengthy and in a short period of time the product can be made available for filing. So there are various departments which are involved in technology transfer. Once the R&D gives the prototype formula and prototype manufacturing process, the role of other departments starts. So in R&D, there are departments like FRD, ARD. ARD means Analytical Research Development Lab is there, which makes the specifications and methods for in-process, finished product, and raw materials, and also packaging materials. Then regulatory affairs is there. Regulatory affairs checks all the documents which are required at the plant for successful execution. Then planning department, these departments are involved in planning of the activities. Then developmental quality assurance, these this department assures that the documents have the required quality. Then commercial and purchase team. So these departments and the team is involved for the purchase of the APIs, raw materials, and the change parts. Also, the purchase team is involved in the purchasing of the packaging materials. Then comes to the finance team and packaging development. So packaging development is involved from starting till the end of the product filing and during the scale up and technology transfer. Then from plant, it will be technology transfer team, PDL team mainly. Then QA, plant QA will be there, plant QC will be there, licensing team will be available for doing the licensing activity as these are the new products for the plant. Then planning, production team will be giving the inputs for scale up and exhibit batches manufacturing. Then purchase team will be there, packaging team will be there who will pack the stability samples. Warehouse will be there for storage and sampling of the materials. Stability team will be there for loading the exhibit batches on stability. Validation team will be there for the method validation and method transfer. And investigation team, if any, they will investigate if any OS or OD is there, or any specific test is missing, or if any product is showing deviation from the test parameters. So these are the various teams involved into the technology transfer and scale up. See here, the involvement of more departments are there. So the responsibility of the R&D is to get the exact requirements for all these departments and to give their inputs to these departments for successful tech transfer. The licenses are required for manufacturing as per the regional regulations, and generally the licenses for manufacturing are taken from the regional FDA authorities. Also, for some of the excipients or solvents like ethanol, acetone, specific licenses are required. These all the licenses are taken from the regional FDA regulations. Then raw material, packaging materials and change parts are required for the execution of the batches. Raw materials involves APIs and excipients, coating materials for tablets, capsule shells for capsules, and so on. Then RMPM, in process finished product specifications. So these specifications are required to be developed by the ADL and FND, and these are provided to the plant teams. Then analytical method verification is required, because this analytical method is developed into the ADL or analytical research lab, and these are transferred to the QC team so that they can analyze the product routinely. If required, method verification is done, validation is must required, and method transfer is required at the site QC. That is, testing method and its transfer is required. Then knowledge transfer to TT or PDL and training for the process. Also CPPs. CPPs are nothing but the critical process parameters, because these products are new to the PDL team, new to the production team. That's why the R&D team should transfer their knowledge about the product gained during the research and development to the PDL team, TT team, production team, and the departments which are working on the new products. So training for the manufacturing process is required, and the critical process parameters are required to be discussed. Then documents transfer is required. Whatever the documents are there for formulation R&D, ADL, packaging development team, all these documents are required to be shared. Equipments and their CPPs are required to be shared to the team. Equipment occupancies are required to be calculated. Sampling requirements required to be discussed, and protocols are required for studies of the scale up and EB batches. Then here I have shown the requirement of discussion of the CMS, CPPs and CQAs. See, CMS are the critical material attributes. CPPs are the critical process attributes or parameters, which these two involves the process, and because of these two parameters, CMS and CPPs, the CQA of the product will be affected. That's why the CMS and CPPs are required to be discussed, and the training required is to be given to the plant team. Then coming to the cleaning validation, MACO and swab limits. So these MACO limits are required to be given, and these are calculated based on the batch size and the least or most potent API manufactured into the production line. So separate MACO value is required to be calculated based on the product batch size and swab limits are decided. So this cleaning validation is nowadays a very important part of the tech transfer and scale up. So that is required to be made available before the product starts at the manufacturing site. Then document completion of BMR BPR scale up reports is required. TT report for scale up batches, that is process challenge study results. So here in the scale up the process is challenged. Like if you take an example of blending process, so if from the R&D blending time is given as 10 minutes, so below the 10 minutes time is challenged for the blending process, like 3 minutes, 5 minutes or 7 minutes, and at last the 10 minutes to finalize the working ranges of the process parameters. Samples for pilot B if plant. Sometimes some of the development teams require sample for pilot B from the scale up, because these batches are made on the actual production scale equipments. So these pilot B samples are taken if required for the pilot B studies. Then the after completion of the successful scale up, transfer process qualification and validation starts. This is after the successful scale up. Then 3 exhibit batches are taken for process qualification and validation to check the robustness and reproducibility of the manufacturing process. See here, many people get confused between the process qualification batches and validation batches. So if in future there is no requirement of the increase in the scale up from EB batches, the EB batches same can be taken for process qualification and validation. And if there is an increase in the batch size for production level, that time again the validation is required to be done. These 3 exhibit batches are made for pivotal stability studies, and minimum 3 batches are required. The exact requirement and the reasons behind these 3 exhibit batches is available in the separate video on this channel. So I request to go there and to watch that video to have complete understanding of the why 3 exhibit batches are required. Then pivotal B studies are done from the 3 exhibit batches. One exhibit batch is selected and pivotal B studies are done. Then after getting the successful stability results, the regulatory filing is done. After regulatory filing, the regulatory authority may issue query to the R&D team, plant team, and as per the query, this query is circulated throughout the departments, and each and every responsible department gives the query response. Then after query response completion, the product get approved from the regulatory authorities. The regulatory authority may be FDA or EMA or any other to which the regulatory filing was done. Then coming to the manufacturing process. Manufacturing process involves shifting, blending and lubrication, capsule filling and packaging. So very simple example I have taken here for the easy understanding. See, the shifting sieves and order of shifting will be very important. The shifting sieves, these shifting sieves are the change part of the machine, and order of shifting will be CPP. Sometimes co-shifting is done, so that co-shifting will have impact on the proper shifting and mixing of the excipient. Then coming to the blending and lubrication. So in this blending time, for example 10 minutes, and lubrication time for example 5 minutes can be given. The blending time and RPM will be critical process parameters, and these are challenged and evaluated during the scale up. Then order of addition, how the materials are added into the blender are important for blending and lubrication. Also, it is very important for proper mixing of the ingredients. Sometimes in blending and lubrication, sandwich blending is done. For sandwich blending, some of the excipients are added, and then the API is added, and after API addition the remaining excipients are added so that the mixing will be uniform. Then capsule filling. In capsule filling, the dosing disc thickness, machine speed and tamping will be very important. See here, the dosing disc thickness will be decided based on the trials taken in R&D, and also sometimes more than 2-3 dosing disc thickness are evaluated for scale up, and dosing disc thickness is a fixed parameter and that is not a CPP. Many of the people get confused between the CPP and the machine change part. So always remember that dosing disc thickness is not a CPP. It is a machine change part. Like punch tooling is the change part for tablet compression. Then in capsule filling, machine speed will be there and tamping will be there as a critical process parameters. Then coming to the packaging. For packaging, the in process pack for dispensing, in process pack for keeping the blend, finished product, and also the filled capsule and empty capsule will be there. Then FPPM, these are FPPM means the finished product packaging material CMAs. That means if you are packaging the capsule into alu blister, then thickness of the alu foil will be critical material attribute. Then CPPs will be machine speed and the sealing temperatures. Sealing temperature and forming temperature depending on the packaging. So these will be the CPPs. So I hope you might have got good understanding of the topic of scale up and technology transfer. In this video, we have covered scale up and technology transfer requirements, which are the departments involved, what are their implications, and we have seen an example of capsule formulation scale up. So in this video series, we will see the other examples of tablet capsules, hot melt extrusion, spray drying, and many more. So keep watching the Pharma Learning in-depth channel. Please do like, share and subscribe to the channel. Thank you for watching.